Hello, everybody, and welcome back to Creation Myths. Looks like everything is up and running. I should see everything live in the YouTube window in just a second. I hope I do, and I do, and see the will, and it looks like everything's good. So, welcome back, everybody, for part three in this three-part series in which we are not talking about creationism at all, which is kind of nice to talk about real science for a change. So here we are today talking about how evolution explains cancer. And we are going to explain basically how and why cancer develops in an evolutionary context, which you can explain the kind of nuts and bolts, like what mutations lead up to cancer occurring, but you can't actually understand the underlying why does cancer happen without understanding the evolutionary dynamics behind it, and that's what we're going to talk about right now. So before we get going, I just want to say that this topic builds heavily on what we've covered over the previous 2 weeks. So remember, 2 weeks ago, we talked about the evolution of virulence in pathogens. And then last week, we talked about the evolution of altruism. So this topic builds on those discussions, and this will not make sense if you haven't already watched those. So if you're watching this, but you haven't seen parts 1 and 2, go check out parts 1 and 2. I'm going to try to remember to link them down below, but I'm going to try to do that. I'll make a playlist. So check out part 1 on virulence, then part 2 on altruism. Then you can enjoy this part 3 on cancer. This will make a lot more sense once you watch those other two. Basically, what I'm saying is that this topic exists in the context of what came before. So make sure you go back and check out that stuff. So with that out of the way, let's talk about how evolution explains cancer. Now, as we get into this stuff, I want to make it very clear. We are going to be talking about a couple of concepts here in ways you may not be familiar with. We're going to twist some things around in ways that might at first be counterintuitive. That's okay. Just bear with me. I promise you there's a reason why we're going to look at things the way we are. So just be aware, we're going to turn some things on their heads in terms of how we typically think about some concepts that might be familiar. We're just going to think about them in very different ways today. And I'm really excited for this. This is super cool what we're going to do today. So the first thing we are going to talk about today is something really important to lay the foundation. So we're going to do our outcomes first, like we always do. I almost forgot about our outcomes. Here are the outcomes for this topic. And if you're just joining us and you haven't seen parts 1 and 2, I'm structuring this like my classes, which means we're going to have learning outcomes, right? We have specific student learning outcomes, things I want students to be able to do with this material. And it's always verbs. It's not just like knowing a thing. It's you have to be able to do something with that information. So for today, our first outcome is to sequence selection for multicellularity, basically be able to explain how that would actually happen because it's more complicated than you would think. Then we have to sequence how cancer develops, kind of in the proximate sense, like mutations happen and that leads to whatever, right? And we'll talk about it. And then the third thing is the really important one. Compare and contrast how opposing selective pressures affect virulence in cancer. Right. And there's one of those words that we talked about previously, right? So we're gonna build on what we talked about 2 weeks ago to make that third outcome make sense. So we're gonna get there. So the first thing you can see on this list is multicellularity. So let's talk for a few minutes about multicellularity. Then we'll layer on top of that. We'll actually talk a bit about cancer. So I'm just we're gonna just scratch the surface of multicellularity here. But simplest definition imaginable, multicellularity is when you have an individual organism made of many cells that are mutually dependent upon each other. And that little bit in the middle, mutually dependent, is really important. That means that it's not just a colony, right? A colony of cells are a bunch of cells that live together, but you could break one off and it could go off and live on its own, and that's fine. That's not true multicellularity. Multicellularity requires interdependence, right? Your skin cells need all your other cells. Your muscle cells need all your other cells. Your nervous system needs all the rest of your body to work, right? It's mutually dependent on each other. Now, I think for this, there's a better way to think about multicellularity, right? This is the typical way we think about multicellularity, right? We have, in a human, it's like trillions of cells. You have a bunch of cells, trillions of them all working together, and we think that those trillions of cells form a single individual. But to make sense for what we're talking about here, I don't want you to think about multicellularity this way. For tonight, this is wrong. Instead, this is the first kind of difference in how I want you to think about things. I want you to think about multicellularity and multicellular individuals as populations of trillions of individual cells. Okay, so we're looking at trillions of cells in a population that make up an individual. So instead of me being Dr. Dan is a population of trillions of individual cells working together, right? In a mutualistic kind of way. All right, so that is our first kind of thing for tonight. I just want to change the way you think about multicellularity. I want you to think about multicellularity as a population rather than an individual. Now, this leads to some really interesting questions and some different ways of thinking about biological entities. Because let's talk for a minute about how multicellularity evolves, right? The basic pathway is like this. You have a group of cells that are operating together like in a colonial kind of way, right? You have a cluster of cells, and they're going to specialize. So some of those cells start to do specific jobs. That means they can do those jobs more efficiently, right? If a cell can do one specific job and then rely on other cells to do other jobs, each cell can optimize for each specific function that it has to do. That means that you're going to increase your fitness, right? If you have an individual that can do each of its functions more efficiently than an individual where all of the cells have to do everything and they have to be generalists and they can't specialize, right? That individual who has the specialization is going to be a better competitor for resources, right? And that leads to higher fitness and the propagation of those multicellular traits, right? Cell specialization and interdependence. Now, that's a very simple process to explain and to even observe. We could directly observe that process happening in the lab. But that's not what I want to think about tonight when I talk about how multicellularity evolves. Because this does lead to an interesting question. We say that that leads to higher fitness because the organism is operating at higher efficiency. But higher fitness for who? Think about an individual, right? You're made of skin and muscles and neurons and a bunch of other things. And the vast majority of those components don't reproduce, right? Or in other words, thinking about an individual as a population, the vast majority of the members of that population don't reproduce. The germline reproduces, but all the rest of the cells and tissues in your body don't. So, like, what's their fitness? What's the fitness of one of your muscle cells or one of your skin cells? What is their genetic contribution to the next generation? The answer is zero. Right? Your muscles don't contribute any genetic material to your kids or your grandkids. Zero. Your neurons, your skin, your liver, your stomach, your epithelium, right? None of it. None of those things, important as they are for your survival, contribute any genetic material to the next generation. Their direct fitness is zero. So how does multicellularity evolve? How can there be selection for traits that result in a fitness of zero for most of the individuals who exhibit those traits? Because again, we're thinking about a multicellular individual as a population. And if most of the individuals of that population are not contributing any genetic material to the next generation, how do you select for that? Shouldn't they rebel against that system so that they can have some fitness, right? Only your germline cells, a minuscule amount of the tissue in your body, has any potential fitness at all. The rest of the members of that population don't. So, what can explain this mutualist arrangement where most of your cells just go along with it, even though their direct fitness is zero? Well, some of you are probably seeing where I'm going with this because I've used the phrase direct fitness a couple of times, and that's a bit of a giveaway. Okay, so remember altruism? Remember what we talked about last time when we talked about the evolution of altruistic behavior? I'm going to do a behavior that benefits the individuals around me. It's going to hurt me and my fitness, but it's going to benefit their fitness. And we talked about how this concept called indirect fitness could help us explain that. Now, what about the indirect fitness component to this equation, right? We talked about how muscles and skin and everything have a direct fitness of zero, but what about their indirect fitness? Well, the thing is, you have to remember, indirect fitness is context dependent. It's based on relatedness. So think again about a multicellular individual. How closely related are all of the cells in the population that is you? Well, we started with a zygote, right? We all start with a single cell that's a zygote. That's sperm and egg fused. We get a zygote. And then that zygote divides via mitosis, which is an asexual process to the adult, right? Zygote to adult. There's no recombination. There's no shuffling up and switching stuff around. It's an asexual process. So how closely related are all of those cells in that multicellular, I'm going to say in that multicellular population, right? The answer is that they're clones. They are 100% genetically identical. Now, I know it's not perfectly 100% because mutations happen, but it's like really close to 100%. If you're talking about a handful of mutations that are going to occur, over the course of however many cell divisions from zygote to adult, yeah, it's not going to be 1.0000000, however many, but it's going to be 99.9 or 0.999999, right? It's going to be very closely related, okay? Right, in terms of just the genetic similarity between the genomes of any two cells in your body, right? Because they are clones, right? It's one zygote grows asexually into the rest of that population. So everybody following me so far? So we have a population of individuals who are all clones of each other. And this is really important when it comes to selecting for altruistic behavior, because who remembers the ground squirrels from last time? The whole thing with the ground squirrels was the individuals who were more closely related to all of the other individuals in the population, the adult females, they tended to make the most alarm calls. They tended to be the most altruistic. And we're going to see that exact same pattern here, except who's most related to everybody else? Everybody. Everyone's the same. They're all clones, right? So that's what allows us to explain this seemingly unexplainable behavior in multicellularity. Okay, so let's revisit this concept of inclusive fitness. So inclusive fitness is the combination of two things: your direct fitness, your alleles in your kids in the next generation, plus your indirect fitness, alleles that you have represented in the next generation through other individuals' offspring. And normally those other individuals that share some of your alleles, they're going to be close relatives, right? So we call selection for this type of thing, it's kin selection, right? You're selecting for things that benefit your kin, and that promotes your own indirect fitness because it increases the representation of your alleles that you possess in the next generation. And this inequality is what explains this, right? This is R is the coefficient of relatedness times B, the indirect benefit, must be greater than the direct cost. If that is the case, then you can select for altruistic behavior. This is what we did last time, right? We're just building on this to explain multicellularity. Is everybody following along with me? This is what we did last time. Now we're going to use this concept to explain multicellularity. If you have any questions, if you're not following, put the questions in the chat and I will see them. I got it open here. So looking at multicellular individuals in this equation, here's the key thing. Everyone's coefficient of relatedness is 1, right? All of your cells, the coefficient of relatedness with all of your other cells is 1 because they're all descended from a single cell asexually, right? So it's zygote to 2 cells to 4 cells to 8 cells, all the way down to trillions of cells. There's no other parents coming in. There's no recombination. Therefore, the coefficient of relatedness across all of the cells in this population, right, that is a multicellular individual is 1. That means in terms of selection and fitness, there is no difference if your skin cells or your germline cells reproduce. R equals 1. The coefficient of relatedness is 1. So selection for the alleles is equivalent, no matter which cells are actually doing the reproducing, right? It doesn't matter which cells are actually directly contributing those alleles to the next generation. It's equivalent in terms of the population genetics math and the benefit of that transmission of alleles vertically to the next generation. Everybody benefits equally in terms of the fitness because the coefficient of relatedness is 1. In effect, this means there is no distinction between direct and indirect fitness in this situation. They are mathematically equivalent, and that means skin cells don't care if they don't reproduce. The germline cells reproduce, and that selectively, in terms of the actual selective pressures operating, works exactly the same way. Does everyone see how that works? Is everybody on board with this? I know this is a very different way of thinking about multicellularity, but I promise you this is going to be the foundation that allows us to explain what's going on with cancer. And I will tell you, some of you are following this. Like I see Andrew here, too many mutations, too much genetic difference leads to cancer. And then just now, except if you're cancer, if you're a cancer cell. Andrew, I will tell you, you see where this is going. You are following along. You see exactly where this is going. How's everybody doing? Are we okay? And it's always weird when I ask that because there's a delay, but okay, I see people saying we're making sense so far. If you got questions, stick them in there. I'll catch them. Let's keep going because we have done the first thing we need to do, which is just explain like how multicellularity works. Okay, so now let me show you this summary. This is the 30,000 foot view here. Multicellularity works when you treat each individual like a population where every member has a coefficient of relatedness of 1 to every other member. So you can look at neurons in the brain, you can look at muscles in the heart, you can look at the gametes from the germline. They all have the same inclusive fitness. And that's how we can select for multicellularity, despite the fact that it really does represent an extreme form of altruism exhibited by most of the cells that are participating in multicellularity. And we have this comment here from Sir Derpier Mister. So essentially, as long as one type of cell reproduces, everybody wins. As long as the coefficient of relatedness is 1 across the population, then yes, exactly, right? Because genetically, it doesn't matter which cell it is. Everyone's related, everyone's identical. So it doesn't matter who directly contributes as long as someone does. Yeah, exactly. So let us continue on. Now we can talk about cancer, right? That was just our foundation. So now let's go to the actual topic for tonight. Now, as you all probably know, cancer is uncontrolled cell growth within a multicellular individual. So you have one and eventually more cells that are growing and dividing uncontrollably, and they take over their local area. They can spread throughout the body, cause problems, right? High virulence ultimately cause death, right? That's the idea with cancer. And this occurs due to the accumulation of mutations in the cancer cells that cause them to escape all different kinds of regulation that normally keep them kind of under wraps doing their specific job within the context of that multicellular individual. So here are just some of the things that typically get broken or disrupted in the context of cancer. So you have genes that are called proto-oncogenes. These are genes that regulate parts of the cell cycle. These are genes that basically say, go. They say, go to the next step, divide, do mitosis, make more cells. Proto-oncogenes in their default state are off, and then they are turned on at very specific stages in the cell cycle. But most of the time, their default state is off. Tumor suppressor genes are kind of the opposite. Tumor suppressor genes, as the name implies, are genes that suppress cell division. Now, that means for those cells, they're actually on most of the time, and then periodically, at very specific times in the cell cycle, they're going to be turned off. So you need to have both of those types of genes typically need to be mutated in order for cancer to happen. You need to lose the regulation of the cell cycle. You also typically have the loss of regulation that has to do with something called context dependence. This just means you have to be attached to something else. So there are two types of context dependence that typically get disrupted in cancer. First, you have context-dependent survival, meaning if you're not attached to something else, you die. Cancer gets away from that usually. Usually cancer cells, at least in later stages, are able to break off from the other cells and tissues that they're attached to and continue growing and propagating and surviving. The other form of contact dependence that is usually disrupted in cancer is called contact-dependent growth inhibition. That means that as cells divide, if they bump into another layer of cells, they tend to stop, right? You detect that you're pushing up against something, and that is a signal that says, okay, far enough, stop growing now, stop replicating, right? Because you could damage the other thing. And then something else that tends to get disrupted is the process of apoptosis. Apoptosis is programmed cell death. So it's when a cell either internally detects like mutations or damage, or it could be another cell that triggers it. It's going to cause that cell to basically break itself down in such a way that actually benefits the cells around it. So it'll actually kind of like package itself up, and the other cells around it get like lipids and proteins and stuff that they can then use. Apoptosis is a very orderly process, and it's a highly regulated process. And it's very different from just like cell lysis, when a cell just like bursts. That's bad for the cells around it. Apoptosis is good, and it's a really important part of actually regulating development, right? And then the last thing on this list here is just resource acquisition, which is basically like a lot of tumor cells actually promote the growth of blood vessels. And that basically causes them to get more resources at the expense of the other tissues in the immediate vicinity, right? So it's all about escaping the inherent regulation that these cells are being subjected to and kind of breaking out of that agreement that they have with all the other cells in the multicellular context, right? And kind of going out on their own and just taking over. That's what's going on with cancer cells. Now, this leads to selection for those traits, because this figure, I think, does a really good job kind of illustrating this. When you have cells that are less regulated in terms of their growth, their cell division, their cell cycle, right, then those cells are going to grow and replicate faster than the cells around them, which means they're going to outcompete the cells around them. So what's happening is in your body, right, the cancer cells are under selection compared to the non-cancerous cells that surround them. Okay, and this figure shows this where you have a series of mutations occurring that lead to faster growth in these tumor cells, right? So here we have a group of cells that's growing faster, and then here we have an additional mutation and an additional mutation and more. So it's this group of cells in red right here are the fastest growing cells in this tissue. So who's going to take over that tissue? It's the cancerous cells. That means that whatever function these other cells are doing is going to get disrupted, right? That's why this causes a problem. Okay, so selection in the very small scale is going to favor the accumulation of these mutations that lead to cancerous traits. Is everyone okay with that concept? Again, we're looking really small scale first, but let's just get that down, and then we'll zoom out and we'll see more kind of big, big picture evolutionary basis for this. Okay, so I don't want anybody to like read this. I just want to show you, the steps in the pathway. This is from a paper from 2003, and it shows the steps in the pathways, the mutational steps in the pathways for different types of cancer. And what you can see here is it's not one or two mutations. It is a lot of mutations that are accumulating here. And this box all the way at the end is a not complete list of the either regions in the genome or specific genes that have to experience mutations in order for cancerous traits to develop. Okay, and there's lots of different pathways to get there, but all of them involve mutations in many, many different traits related to the things that I mentioned a few minutes ago, and also stuff that I just didn't mention because we don't have to go through all of the mutations that happen. We'll be here all night. But takeaway here, a lot of mutations accumulate within these cancerous cells. One of the reasons for that is these cells are replicating faster. Every time you replicate, you copy your DNA. The more times you copy your DNA, the more opportunities you have for errors. And on top of that, another characteristic of cancer cells that I didn't mention in that list a few minutes ago is you often see the loss of various error correcting mechanisms for like DNA replication. So it's not just that more replications give you more mutations because it's however many mutations per replication. You're also getting more mutations every time you replicate because you're losing some of that proofreading, error correcting stuff that the non-cancerous cells can do. So the takeaway here, again, is not the specific stuff in this figure. The takeaway is that it's a lot of mutations. And this figure does an even better job conveying just how many mutations it is, because it's not just a few mutations, it's many mutations. And it's not just point mutations where you have, an A turns into a G in a specific gene. It's large scale structural changes to the genome, the actual chromosomes. So here in this figure, on the left, we have a typical human karyotype right here, and each pair of chromosomes is color coded. So you've got 1 through 22, your autosomes, and then X and Y down here. Here is a karyotype from a cancerous cell. You can see you have all kinds of duplications and translocations all over the place, and you have a bunch of what are called clonal and non-clonal markers, which are basically just assemblages of like fragments of chromosomes that get stuck together, and we can't classify them as this or that specific chromosome because they just have so many different pieces mixed and matched together, right? Obviously, this genome is very, very different from this genome, right? The genome on the right is very, very different from the genome on the left. In fact, you might even say that the genome on the right is not so related to the genome on the left anymore in terms of its similarity, right? Base by base. Now, this is a really interesting question here. Is B cancer the default mode and those regulatory mechanisms suppress that? I think the way I would think about this is a fantastic question. This is the kind of question I would love to get like when I'm doing this in class for real. This is great. So this is DM, we be doing evoluting wing. Sorry, there you go. Thank you for this question. This is great. The way I would think about this is that multicellularity suppresses the kind of default looking out for yourself mode that most things are operating under, right? So you look at unicellular organisms, right? It's every cell for themselves, basically. And multicellularity suppresses that, right? The cells kind of reach an agreement, as we talked about a few minutes ago, due to their relatedness. We're going to say, you know what? I'm just going to help everything work. You're going to actually reproduce. But because we're clones of each other, in terms of the selective pressures, we're not competing with each other. If you succeed, I succeed, right? It's that regime where you have a coefficient of relatedness of 1 throughout the individual leads to the situation where I would say you're selecting against doing this, right? It's a better strategy to be a mutualist than to be cancer, right? That's why I would answer this question. I think the default mode pre-multicellular is everyone for themselves. Multicellularity is, we live in a society, we're going to look out for each other. And then cancer is going back to that pre-multicellular, every cell for itself kind of mindset. I hope that answered your question. That's how I tend to think of this. There's a really, really good question. Thank you for that question. Okay, so let's continue. Because now let's put this in the context of multicellularity, right? Because we just talked about, and that question kind of got at it, about like selection for multicellularity. And we talked about how it's beneficial, both in terms of like you can be more efficient because your cells specialize. That gives you a competitive advantage, increases your fitness. And also because everybody is a clone, right? R equals 1, then that fitness improvement due to being multicellular and efficient applies to everybody, even the cells that aren't directly reproducing. Now, cancer cells are saying, no, we're done with that. This is a breach of contract, right? We are abandoning that agreement with all the other cells, and the cancer cells are no longer acting altruistically. So think about like those two figures I just showed you, right? With all the mutations that accumulate and all the differences that can happen at the genome scale, what's happening to the relatedness of those cancer cells? Well, now that R is less than 1, right? They're no longer genetically identical to all the other cells in the body. Does everyone see that? I know Andrew sees that. He's been spoiling this slide for the whole time, and I swear we didn't talk about this ahead of time. He just saw this coming from a mile away. Apparently my foreshadowing was extremely obvious. Everyone else, is everyone okay with how we just got here, right? Cancer cells now, by accumulating tons of mutations, growing out of control, they are breaking out of this multicellular mutualism because R is no longer 1. It's way less than 1 at this point. That means the math doesn't work anymore. If the germline cells successfully reproduce, how does that affect the indirect fitness of the cancer cells? It doesn't help them anymore, right? It's not a one-to-one thing with their fitness anymore, which means we can't treat cancer like it's part of a multicellular individual or a member of that population of cells that makes up the multicellular individual. That paradigm doesn't work for cancer anymore. Instead, to explain cancer, we have to treat it like a parasite infecting a host. That's the context in which the subsequent evolutionary dynamics of cancer are going to work. So that's what we're going to talk about now. So now that we've explained multicellularity and how cancer breaks out of that, now we can explain, okay, why is cancer virulent in that context as a parasite rather than as a member of a multicellular population? Okay, so now we're changing the framing again, and I know there's a lot of like back and forth in terms of how we're thinking about this, but now we're changing the framing here, and we're going to talk about cancer in terms of host-parasite relationships rather than a part of a multicellular organism. So let's talk about cancer as a parasite. This is obviously going to build heavily upon the stuff we talked about 2 weeks ago when we talked about the evolution of virulence, right? So once cancer gets going, once that snowball starts tumbling down that hill, cancer is going to behave like a parasite. It's going to experience selection due to the competition within its host. And that selection is basically the kind of selection that pathogens experience. So remember from 2 weeks ago, we talked about kind of two selection dynamics that can occur in pathogens. You can have intra-host competition. That's where you have a population of pathogens or multiple species of pathogen even within a single host, and they're competing with each other to take over within that host environment. But that's only part of the story because pathogens also transmit. So you also have inter-host competition, which is illustrated here, where the pathogens in different hosts are competing with each other to infect the available hosts in the population. Okay, so this is what's going to help us understand the selection dynamics that affect cancer once cancer appears in a multicellular organism. We want to think about it like a parasite. So here's how the selection for cancer works. Tumor cells experience strong intra-host competition, right? Tumor cells are competing with all the other cells in your body and this is really important, with each other within the tumor. So sure, it's easy to outcompete the non-cancerous cells, but competing with the other tumor cells is really hard because what are tumor cells really good at? Growing fast, replicating, making more of themselves. So if you're going to be experiencing selection due to competition with the other cells in that tumor, you're selecting for things like faster replication, outcompeting nearby cells, spreading to new locations. Basically, you're selecting for traits that promote infectivity within your current host. This is going to result in higher and higher virulence, right? This is going to be a positive feedback loop, like we talked about 2 weeks ago, that leads to higher and higher virulence. Okay, is everybody okay with that in chat? If you're not following, ask questions. Okay, want to make sure everyone's following along here. I'm delighted to see there's 31 of you here as I say this, and like that's awesome. Thank you. I hope you're all enjoying this. And if anyone has any questions, stick it in there and I'll answer them. This is great. So is this a good long-term strategy, right? Think about this as a parasite. If you're a parasite and your host dies, what happens to your fitness as a parasite? You know, if you're a tapeworm or, the plasmodium that causes malaria, right? And your host dies, what happens to your future fitness? It's zero. You're out of luck, right? So in typical parasites, in addition to intra-host competition, they also experience inter-host competition, right? Which imposes selection to transmit to a subsequent host. Because if you take over your current host and then your host dies before you transmit, it doesn't matter if you took over your current host, your contribution to the next generation is still going to be zero. So you have this other force that pushes back, right? The inter-host competition reins in the virulence so you don't, fail to transmit, at least in some cases, not always, but in some cases. But in cancer, that doesn't happen, right? Because there's no transmission in cancer. So there's no check on the virulence. So those tumor cells are under selection and it's a positive feedback loop to get more and more infective within that single host, which means you're going to get more and more virulent. You're going to take over the region where you appeared. You're going to be able to break off and spread throughout the body and take over those other parts of the body. And ultimately, that's going to kill your host because there's no transmission. There's no way to select against that positive feedback loop of virulence. That's why cancer progresses and why early detection and treatment matters so much. If you catch it early enough, then you can cut off that feedback loop, right? But once you start going down that feedback loop, it becomes much, much harder, right? So this is the key here, and this is where we could do a little, public health, public service announcement. Like whatever age you are, whatever screenings are appropriate for your age, get those screenings, right? You catch this stuff early, you don't have to worry about like all those mutations building up because you can catch it at the front end. And I love the fact that everybody knows where this is going. I can't tell you how happy the comments in the chat this is making me right now. So we've got Mark Reed. Suppose it doesn't matter as long as you pass along your genetic material. Sorry, spoilers. And then we got Andrew. What about so-called transmissible cancers? And then we've got CTRO, except Tasmanian devils. Sorry, there's one. I was thinking about the transmissible dog cancer, right? You all know exactly where this is going. This makes me so happy. I feel like I've done my job. This is great. And Mr. Anderson, also, I lost it, but you mentioned transmissible cancers all the way back at the beginning. So yes, thank you. I don't have, nope, not that one. Hang on. Here we go. I don't have the comment, but yes, Mr. Anderson called it all the way at the top. Yep, is great. Okay, so let's continue. Let's close this loop here. Let's explain these dynamics, right? Because again, the point here is cancer doesn't transmit. We get runaway selection for transmissible virulence in cancers, except there are transmissible cancers. Now, I want to be clear about this. We are not talking about cancer caused by a virus, right? We're not talking about something like cervical cancer caused by human papillomavirus, which is almost entirely preventable because over 99% of cases are caused by HPV. If you are of the appropriate age, get your HPV vaccination. In places that have a lot of HPV vaccinations, basically eliminated cervical cancer, do that. Epstein-Barr virus is another one that's associated with a bunch of cancers. HIV is associated with certain cancers. That's not what we're talking about. That's like a whole other thing. We could spend, 45 minutes, we could spend 80 minutes just on that. We're not talking about that. Instead, what we're talking about here are the very few cancers that can actually jump. The cancerous cells can jump from one host to another. And there are a handful of these that are known, 6, maybe 8, maybe more at this point, but a handful of transmissible cancers have been documented. And we're going to look at 2 to see how the dynamics that drive the evolution of virulence that we talked about previously apply to cancer and how transmissible cancers actually illustrate that. Okay, so let's talk first about canine transmissible venereal tumor. Now, this is a temporary, non-fatal growth that appears on the genitals of dogs. It is transmitted from individual to individual during mating, and it is extremely ancient. It actually predates the domestication of dogs. You can find this stuff in modern gray wolves, as well as domesticated dogs. Now, what's going on here is this cancer basically behaves like a parasite, right? It is essentially a parasite, and it has to transmit, and it has a fairly limited window where it can transmit. So what I think happened here in the evolution of CTVT is that you have selection for lower virulence to promote the transmission of these cells. And not to be, too graphic about it, but this is a tumor that grows on genitals that requires mating in order to transmit. So if it damages those tissues or causes pain, I think we can safely say it's not going to efficiently transmit, right? So we're going to see selection for low virulence that's going to facilitate transmission in these particular for these particular cancer cells, right? So this has been going on from tens of thousands of years. However, I'm not sure exactly what the dating is, but it's at least tens of thousands of years, right? So that's the ancient example. Let's compare that to what's going on with a modern example. And some of you called it in the chat here. Devil-faced tumor disease is the transmissible cancer that affects Tasmanian devils. This is extremely recent. The first documented cases appeared in 1996. And until the mid-teens, the mid-20 teens, I think it was, we had a 100% fatality rate. So what would happen is the Tasmanian devils, they kind of bite and scratch each other. And if one of them had the tumor on its face, those tumor cells would break off and they'd get into the wounds on the other Tasmanian devil. And then they would kind of grow and take over their face. And I know, poor devil, right? It's very sad. I know, poor devil. I know, Tasmanian devils are adorable. And so it would kind of take over its face, and ultimately it would prevent them from eating and also from breathing. So it was 100% fatality rate. There's a whole other thing that's going on here with life history traits and the time it takes them to reach sexual maturity and extrinsic mortality. That's a whole other conversation. Maybe I'll do a stream about that at some point. But that's not the topic for today. Today, we just want to focus on the dynamics of this tumor, because like I said, this was actually had people worried that it was going to drive Tasmanian devils to extinction until, and I don't know the exact year on this, but I remember it happening when I was in grad school. So early to mid 2010s is when this happened, that we started documenting resistance in some populations of Tasmanian devils, where the tumor would grow and then it would go away. It seems like the immune system would defeat that tumor. And it tends to be described as resistance to devil-faced tumor disease. But I also want to hypothesize that what I think is going on here is there's also selection operating on the tumor itself to be less virulent. Because if that cancer burns through a population and kills 90 plus percent members of that population, it's going to have a much harder time spreading when you've reduced the population by that much. So now we're going to see selection in those transmissible tumor cells to actually not kill their hosts, because the ones that allow their host to survive for a longer period of time, those are going to be more likely to be able to transmit. So I think what's going on here is that we are watching this transmissible cancer adapt to an environment where it's being subject to inter-host competition as kind of the bottleneck step in its transmission cycle rather than intra-host competition. And that's actually imposing selection for lower virulence. And that's why it's no longer fatal in 100% of cases. Right? I think that might be what's going on here. This is a hypothesis. I'm not out there studying devil-faced tumor disease and like sequencing the genomes of tumor cells. I'm not doing any of this work. So I don't know. I don't have a basis, data to support this hypothesis. But I think when we do get those data, I think that may be what we see. It's going to be resistance in the Tasmanian devils, but also we're going to see genetic changes in the tumors that facilitate transmission, and part of that is going to be lower virulence, right? So that's a really interesting illustration of how we can have a cancer that once we add transmission to the equation, it starts behaving exactly like a parasite. So I think that's what's going to happen there. It'd be really cool. I'm periodically I kind of look into this, and it'd be really interesting to see where that research goes over the next decade or so. So that brings us to the end of this topic, right? We're talking about cancer and the evolutionary dynamics of cancer. So cancer is a disease of uncontrolled cell growth and division in a multicellular organism. Multicellularity, as we talked about, it's possible due to kin selection plus high relatedness. So it doesn't matter which cells reproduce, genetically it's equivalent. So you can have selection for this very strong altruistic behavior in your multicellular population that is you. Now, cancerous cells experience lots of mutations that cause them to outcompete non-cancerous cells. They break that kind of mutualist agreement because they're now less related, right? And they impose parasite-like selective pressures, right? They experience parasite-like selective pressures. Without transmission, that means that cancer only experiences selection that promotes higher virulence, which explains why cancer gets worse and worse and worse, typically when it happens, right? It doesn't spontaneously go away. So that's it, everybody. That is our chat on the evolutionary dynamics of cancer. I hoped that caused you to think about things like multicellularity and what cancer is in a slightly different way. Now, we did have one super chat in here. I want to make sure I highlight this from Charles Payette. Trillions of me's, terrifying thought, but it's true. You are trillions of you. Just don't think about that too hard. It makes your brain hurt. But yes, and thank you for that super chat. That's very generous. I really appreciate it. Andrew, I did not see your question about this, but if you can find it real fast, I can try to look at it and answer it for you. If anybody else has any questions about this, by all means, please feel free to ask them. And then once we kind of run through the questions, I will call it a night. Do you think like chemistry or nanotechnology more successful to control cancer? Yeah, so you could go like I think it's like a nanotechnology in The Expanse, right? I would love for that to be the case. That would be really cool. This is from Sydney Raptor. Great question. Ultimately, the solution for cancer is going to be germline gene editing or embryonic gene editing with like CRISPR. And we just fill our genome with 20 copies of a tumor suppressor gene called P53. Elephants, this is an aside, but it's a really cool thing. So I'm just going to tell you about it because it's cool. Elephants have a lifetime incidence of cancer of about 3%. compared to about 33% for humans, about 1 in 3 for humans. The difference for that comes down to this gene called P53. P53 is a tumor suppressor gene, and it's mutated in almost every type of human cancer. Elephants, you would expect to experience more cancer because from zygote to adult, it's just they're bigger, it's more cells, it's more cell divisions. So you would expect more mutations, more cancer. So elephants early in their lives would, throughout their evolutionary history, have experienced cancer. So they're under very strong selective pressure to minimize cancer. And the trait that they have selected for is multiple duplications of this gene P53. And here you go from CTRO. I'm surprised you didn't talk about the 52 copies of P53. Yeah, it's a little bit, adjacent to what we were talking about tonight, but maybe I'll do another one on that. I'm like kind of mentally building up these other topics that we can do. And this is a really interesting one, how elephants have tons of copies of P53. And once we get to the point, and it's a when, not an if, but once we get to the point where we can modify the genetics of human embryos, we can just basically immunize individuals against cancer. And it's about 20 copies of P53. And here's the cool thing with this. If you could do it all at once, you could basically eradicate cancer forever, right? Because if once you have those 20 copies of a tumor suppressor gene in the genome, that person's children inherit those 20 copies. So you don't have to keep doing the gene editing generation after generation after generation. You just do it one time, right? You catch everybody. So this is how we're going to beat cancer, right? Some of the immunotherapies we have are basically miraculous compared to cancers from 20 years ago. But this is the way to do it blanket. And if we were to do a Manhattan Project, Apollo Project, a smallpox eradication campaign, once we have the ability to do this, we can effectively get rid of cancer within just a few generations. Will we do that? Not super optimistic about humanity's capacity for, collective action for the common good right now. Let me just put it that way. But we will have the technical capability to do it at some point. While we were chatting about this, got a super chat here. No, here we go. From De El Sendero. This is for $15. Thank you for your generosity. These have been 3 outstanding lectures too. It's very satisfying to know a professor of dance skill is teaching at my alma mater. Thank you. I appreciate that. Thank you. Really appreciate that. Let me see. Here we go. From DM, we do be evolutin wing. I suppose generally less cancer isn't selected for because it doesn't affect reproductive fitness. I'm going to agree with this conditionally, that I'll say across our evolutionary history, right? So historically, right, you go back 200,000-300,000 years, what's the human lifespan during that time? It's about 40 years, right? But when do most cancers occur? Much later in life, right? So there's historically going to be no selection pressure against cancer that doesn't typically appear until you're 60, 70, 80 years old. Whereas for elephants, because again, more cell divisions, right? More opportunities for mutations, more opportunities for cancer, they actually would develop cancer in their typical lifespan, and it would impact fitness, and historically has. And that's why they've experienced selection for all those duplicate copies of P53, right? That's what's going on there. So you're exactly right. It doesn't, or I'll say it hasn't affected fitness in humans, but in other things, elephants and also in whales, right? They're very low incidence of cancer. These are things that are so big that absent the anti-cancer mechanisms they have, they would get cancer earlier in life and it would affect their fitness. That's the dynamic that's at play there. Yeah. Great question. Okay, so this is, I think, a good place to wrap this up. So I just want to, again, say thank you all for being here, spending this time with me, just going through these 3 topics. Like I said, these are sections of classes that I teach in my real life. It's really exciting to get to kind of share a little bit of that with everybody. So before we go, I'll just remind everybody, please hit the like button before you go. Leave a comment. If for some reason you're here, you're hanging out, and you're not subscribed, make sure you subscribe. And share this around. Like science communication is the thing here. I would just want to share this stuff because I think it's cool and I want to get more people interested in it. So if you would all be so kind before you go, I would really appreciate you doing all of those, algorithm and social media friendly things for this. And with that, I'm going to wrap it up and say goodnight, everybody, and really just thank you all again. I had a ton of fun with this little series, and I think there's more stuff I can do along these lines. So I think I'll do this again at some point down the road. We are going to be back after this to more of our typical programming here, but we'll come back to the real science a little bit down the road, I think. Thanks, everybody. Have a great night.